Perfect. So I think we can get started. Today will be a very interesting topic. PhysioLabXR, it's a software platform we built for real-time brain-computer interface. Let's get started. Great. So during the session, if there is any question, please interrupt me. If there any problem with the screen sharing or microphone, please also interrupt me. Yeah. Perfect. Okay. Can everybody see my screen? Okay. So now we're going to get started. Okay. Great. Cool. Okay. So hi everyone. Welcome to the PhysioLabXR PreHackathon workshop. Today I will introduce you a powerful platform for closed-loop BCI applications, and everyone is welcome to ask questions in the chat. One more people joining us. Okay. Cool. Before we start, so here are some resources. The first one is our official website, and you can see all the features about PhysioLabXR and what it can do. Second one is the GitHub, so where you can download and install PhysioLabXR on your own PC. And third one is a documentation page that listed all the detailed features about PhysioLabXR, and there are many demos that you can run on your PC. So before we start, a little bit about myself. I'm a second-year master's student in the computer science department at Columbia University, and I'm the co-founder and lead software engineer of the platform. My research primarily focused on VR, AR, brain-computer interface, deep learning, real-time signal processing, neural imaging, and human-computer interaction. So in today's session, I will introduce what PhysioLabXR is and discuss the main features of the platform, and each feature will be followed by a live demonstration. Afterwards, we will engage in some hands-on activities. Cool. So let's jump into the introduction. Okay. So let's first discuss the motivation that we build the platform. So in many experiments or while building a brain-computer interface or any applications involves real-time data acquisition, it is important to visually inspect the sensor's data as it comes in. So for instance, during a closed-loop brain-computer interface experiment, we might have EEG signals. So here we have the unicorn hybrid black device that streams the EEG signals to our PC. And we may have the eye tracking data from the Vision Pro. And here, those lines represent the pupil size from the eyes. Or stimulus presentation software like Unity that streams the event markers to indicate when the stimulation occurs. So during the experiment, we need to synchronize those data on a same clock and to extract the data that corresponds to a specific event. So here, we have a very simple application. Okay, this may look familiar to some of you. This application is a lab recorder. It was widely used for multi-streams recording, but it only allows you to check the presence of a stream and start and stop the recording with a button press without any additional sanity checks. Sometimes your EEG signal might become very noisy, and you wouldn't realize it until you read the recorded file after the experiment. And LSL is an open source networked middleware ecosystem to stream, receive, and synchronize and record neurophysiological data and behavior data streams acquired from different sensors or hardwares. So basically, this application is built on top of software middleware, which is called LabStream layer. And there are many neuron tech companies are using it, such as OpenBCI, the GTech, and NearX. So all their devices are supporting LabStream layers. So and it's also supported across different languages. So you can use it in C, C++, Java, MATLAB, Python, and what else? Let's see, and C#, which you will use in the Unity. Okay, so let's discuss about a closed loop system. So the process that we develop a brain-computer interface. So to develop a closed loop system, we typically need to first build the paradigm and run pilot tests. Then analyze the data offline. Finally, we recruit participants and evaluate the system with a user study. And automatically, we hope it will lead to some publications, or we can deliver the application to the customers. So during the process, we may want to visually inspect the data in real time. So as it comes in, we can just visualize it immediately. And hopefully we can record the data for analysis after the experiment. And even more, we want to add our customized data processing pipeline. And even more, we want to test the pipeline over and over again with different parameters. So in that case, we need a system that can visualize the data, record the data, and replay the data. And while we replay the data, we can test our signal processing pipeline with different parameters. And eventually, we finish the product or publish the paper. So that's why we have PhysioLabXR. PhysioLabXR is a Python-based open source software platform for developing experiments for neuroscience and human-computer interaction that involves real-time and multimodal physiological data processing and interactive interfaces. So in general, inside PhysioLabXR, you can visualize the data streams in different formats in real time. So here we have the time series, we have the images, we have the spectrogram, which is very useful for audio signal visualization. And we have the bar chart, which is very helpful for the real-time machine learning inference. And you can add your customized data processing signal pipeline. And you can replay the data and record it and replay it. And you can also add a scripting interface to your data processing pipeline. So the scripting is one of the most powerful features in PhysioLabXR, and we will dive deep into this during the session. So here we have some publications built on top of the platform over the last couple of years. And currently we have about 5 or 6 research labs are actively using the platform. in Harvard, Worcester Polytechnic Institute, MIT, and Columbia University. So now let's jump into the main features. Okay. So here is the launching screen of the application, which you will see when you open it. Let's go to the application. And currently I'm running this from the source code. And you can either download the exe file or clone the repository and run it with PyCharm or Visual Studio VS Code, as long as you have the Python and pip on your machine. Okay, great. So at this point, I open the application, and here's the main window of PhysioLabXR. So inside PhysioLabXR, you can add any streams from lab streaming layer, ZMQ, or camera, or audio. So for example, if your computer has a camera on it, or you plug in a microphone, when you start the application, PhysioLabXR will automatically detect those devices and add the device into the drop-down window right here, and you can just simply click it and add it. Okay. And to add the lab stream layer streams, the only thing you have to know is the stream name. So let's have a live demonstration. So currently what I'm showing is how to create a simulated EEG signal right here. So for example, and you can find this script in our repository. Okay. So the name of the stream is dummy 8 channel. Okay. And the sampling rate is 128, and it has 8 channels. Okay. So while run this script, so what this script does is it creates a simulated stream and broadcasts the data into the local network. So as long as your computer is connected to the same router or like a same Wi-Fi, you will be able to receive the same stream on a different platform, different source. Maybe, for example, if I broadcast this stream on my computer with a Python script, you will be able to receive the data on another computer, maybe in C++ or C, as long as you use the lab streaming layer package. So that's a library you can install online. Okay, perfect. So let's stop the stream first. And let's go back to PhysioLabXR. Okay. So to add a stream, the only thing we need to know is the stream name. Okay. And then we can click the add button right here. Perfect. So because I added the stream with the same name before, and I did some format change here, that's why it looks a little bit different. So we will talk about how to group and organize your streams in the next few slides. Okay, perfect. So maybe, yep. Okay. Right. So at this point, I can start the stream. Now you can see it's waiting the stream from the lab stream layer. And if we go back to the stream that creates the lab stream layer in Python, and I can run the script. So at the moment I start the simulated stream, you can see the data comes in. Okay. And here we have 4 channels in the first group and 4 channels in the second group. Perfect. So during the hackathon, you won't use a simulated stream. Instead, you will have the real EEG streams from the devices, like from OpenBCI and GTAC. So there are some examples. And and you can remove the stream with a button click on the top right corner. Okay. And maybe do another example. So for example, I have the stream that's called unicorn hybrid black itself. So imagine that's the EG signal from the unicorn hybrid black headset. So this is a headset you will use during the actual hackathon. Okay. So let's stop it and maybe restart it. Okay, perfect. Now, if I go back and I add the stream here, so for GTAG, let's have 19 channels right here. And change the sampling rates to 250. And we run a simulation again. So here we have the simulated EEG streams for GTAG unicorn device. Right? So maybe let me redo the sharing. Let's share with, sorry. So here we have the simulated EEG streams from the lab stream layer. And we can remove it. So, perfect. Okay. So that's the first feature, and it's also the most important feature in PhysioLabXR. Okay. So here you will have the main window, and after you add a string with the corresponding name, you will have the stream widget for that stream. And the second feature in PhysioLabXR is you can drag and drop channels into different groups, and you can create new groups by selecting those channels. So let's go back to our demo. And so let me redo the simulation. So inside PhysioLabXR, after you add a string, and if you see the label right here turns blue, it means the uh, string has been found on the local network, and you can add it. So there's some channels. So let's change this to 8 channels. So here we have the simulated EEG streams from the lab stream layer. And to regroup the channels. So on the bottom of each string widget, there is a gear shape icon right here, and it's a button after you click it. So it will open this settings window, okay, right here. And so, for example, I want to create a separate EEG group for T3 and FP2. Okay, so those are the channels name in the 1020 uh, EEG headset. Okay, so basically that's a, a name for the EEG channels in the 1020 EEG system. So now I can create a new group. Perfect. So at this point, I created a new group for T3 and FP2 channel right here. So you can see, basically we split those two, we split those 4 channels into 2 groups. Okay. And you can also drag channels from one group to another. So you can separate those visualizations. One of the reasons we are doing this is we want to put the related signal channels into the same group. So for example, for signals from a device, it may contain accelerators and EEG signals, maybe event markers, triggers, and maybe timestamps. So it won't make sense to visualize the timestamp and EEG signals in the same group, and they are in very different scales. So that's why we want to split those channels into different groups. Okay. Great. Perfect. Go back to the presentation. So the third main feature of PhysioLabXR string widget is you can change the sampling rate for each channel. So at the bottom of the settings window, you can change the nominal sampling rate of the string. So for example, if you have a EEG signal from the biosemi, okay, and their device has a very high sampling rate, like 2K sampling rate, you may want to change the nominal sampling rate to match with their actual signal sampling rate. So in that case, you will have the signal in the same time domain. Okay. So for example, the actual sampling rate for this simulated string is 128. It won't make sense. So for example, if I change this to 1000, you will see it goes very slow. Okay. So ideally, we can change the, so here you cannot set the sampling rate to 0, so it will return you a warning. I want a sampling rate that matches with the actual sampling rate. Okay. And at the bottom, you can also change the number of seconds you want to display the signal. So for example, here, if I only want 1 second signal display, I can change this to 1 second. Maybe I want 5 seconds. So here we have a 5-second signal visualization. And you can drag those channels around. And at the bottom right here, you can click the auto scale button. So it will automatically scale the signal for you. Perfect. So skills. Let's go back to the and the first feature in the stream widget is the pop and dock stream windows. So, for example, in a experiment, you may have multiple streams from multiple data sources. Sometimes you will have EEG signals, the eye tracking data, the recording from your camera, and maybe participants' audio information. So this feature right here, it will allow you to pop out the stream widget in your main window and reorganize them into different parts of your monitor. You can move them around. Right. So here's an example. So this is a real experiment in our lab. And so from the screenshot right here, you see we have the ECG that records the cardio information from the participants. And at the bottom, we have the 64 channels EEG signal from the biosemi. And on the right, we have the eye tracking data from the Vario headset. So you can see it right here. So that's a Vario headset. And inside this Vario headset, there are eye trackers behind it. And we also have the event markers from the Unity. And on the bottom right, we have the real-time fixation detections from the scripting interface, which we will further explain maybe in 20 minutes. Yeah. Perfect. And inside PhysioLabXR, you can also visualize the streams in different formats. So at this point, you can visualize time series streams in line chart, bar charts, image, and spectrogram, which is a fast Fourier transform format. So let's go back to the stream right here, and let's dock it back. So at the top right here, so for example, we could visualize it as a pixel map right here. Okay. So let's see. Let's visualize it as a as a 2x2 matrix right here. And I choose to visualize it as a pixel map. Okay. And I can change the display scaling right here, as well as the color map. So I can visualize it in different color maps. Okay. And we can also visualize it as a bar chart. So one of the use cases for bar charts is to visualize the real-time prediction results from the machine learning model. So for example, if you have a real-time classification model that continuously outputs the classification results, this will be very useful. And you can also visualize it as a spectrogram. So this is so what it does is we run a real-time fast Fourier transform for each channel, and here's the output. Perfect. So additionally, uh, you could have you could add your customized data processing modules in PhysioLabXR. So here, if we go to the main window right here, and we click the gear button at the bottom of the uh, stream widget right here, and we are able to add a customized filter to a specific visualization group. Okay. So in the real world scenario, one of the questions is why we need filter. So in the real world scenario, the EEG will be very noisy, okay? And you're gonna be noise from different sources. So for example, you could have the 60 hertz noise from your power source, and you could have uh, noise from the participants' artifacts. So, for example, if they move their head a little bit, or there any type of oscillations in the space, it will be captured by the EEG electrode. So that's the reason why we need the filtering. Okay. So inside PhysioLabXR, you can add a filter to a specific visualization groups. So in the settings window, we provided multiple built-in filters. And the two that will be used most frequently will be the notch filter and Butterworth's bandpass filter. So the notch filter will allow you to remove the noise at a specific frequency. Okay? So for example, in United States, there's always going to be a 60 hertz noise from the power source of your EEG device. Okay? And you may want to add a notch filter to your visualization group to remove that noise. And the second one will be the Butterworth bandpass filter. So let's do a demonstration of this. Let's add a filter right here. Next. So after I add a filter, here we will have a separate widget for filter design. Okay. So now I add a Butterworth bandpass filter. And here I can define the cutoff frequency. So that is the frequency that I want to keep. Okay. So usually for EEG signals, we want 0.5 to maybe let's say 50. Okay. And here the sampling rate, 128. And let's change the order into 4. And maybe set the cutoff to maybe 40. Okay, perfect. And after I designed the filter, I could activate the filter with a button click right here. So this is a very important feature we will use in this hackathon. For the device from OpenBCI and GTAC, the unicorn device. So let's see if I can find it. So for example, this is a unicorn device you will use in the hackathon. And before you do any further signal processing or analysis, make sure you have the notch filter and Butterworth bandpass filter all your EEG channels. It's very important. Okay. And now if I activate the filter, okay. So you can see at this point, I only kept the signal frequency between 0.5 to 40. Maybe it's not very obvious. So because currently I'm sending random data between 0 and 10. Okay, so let's change the cutoff a little bit. Let's only keep the signal from 8 to 10 hertz. Okay, perfect. Now you can see it's perfect sine wave, right? Because currently we only have the signal from 8 to 10 hertz. And furthermore, you could visualize it in the frequency domain. So if you right-click on the visualization group on PhysioLabXR and go to the transforms, and you will be able to visualize every single time series stream in the power spectrogram, which is FFT. So here you can see that's a power spectrogram of those 4 channels. And we got FP1, F7, F8, and T4. And in the power spectrogram, we only maintained the signal from 8 to 10 hertz. And at this point, if I cancel this filter right here, so I deactivate it. Now you can see all the noise comes back, okay? And the frequency is everywhere because I'm sending a random signal. Perfect. So let's visualize it again in a time series. So here we have a time series signal. And at the point, activate the filter. So the harmonic waves, because we only got the signal from 8 to 10 hertz. So yeah. And for more detail about this filtering functionality, you could refer to our website and documentation. So inside the PhysioLabXR documentation, if you go to the features and real-time digital signal processing, you'll be able to see a detailed documentation about it. So on this page, so there are detailed explanation about the parameters for the pre-built filters and how to add the filters and a sample script. So an explanation of the source code, so you can apply it maybe in a separate project. It's open source. And here's an example of the filter. So here we got a signal with mixed with 50 hertz and 100 hertz. And now we apply a low pass filter with cutoff frequency for 70 hertz. Okay, so we only allow the signal with frequency lower than 70 hertz to pass this filter. So on power spectrogram, we could see like 2 frequencies, the 50 and 100. So for more detail about this, we can watch some online tutorial about why it's symmetric for the negative and positive. Basically, we are visualizing it in a radius axis. Okay. And here, after we apply the filter, we can see the output signal only contains the 50 hertz signal. Yeah. And the 100 hertz signal has been removed. Great. Any questions so far? If there is any question, let me see if it's anyone asking questions in the Slack channel. Organizers. No. Okay. Perfect. And a few more tips for the time series visualization in PhysioLabXR. So for example, if you have the 4 channels in a single visualization group, what you can do is you can add a constant offset to every single channel to visualize them in different scales. So for example, at here, I may want to visualize those 4 channels separately. And what I can do is I could add a 20, or let's add a 10. Okay. Now I add a scale of 10 to every single streams. So basically I add a constant to each streams to separate, to visualize them separately. Perfect. And for more detail, you can refer to our documentation and website. Great. And the next feature in PhysioLabXR is the optimized serialization interface for recording high throughput data. Okay, so we natively support our data structure, which is called DAT. So it's a data structure we created for multi-streams recording. It's a dictionary of array of timestamp and optimized for synchronized multi-stream data. But you can also record your experiments in Python Pico or .m, which is from the MATLAB, or CSV, or XDF. So let's jump into the demonstration. And let's cancel filter here. Now you can see the signal comes back. Okay. So let's jump into the recording. On the top, there are 4 different tabs: the stream, recording, replay, and scripting. Okay. So in the scripting tab, in the recording tab, after you click it, you will see the recording interface. And in the recording options, you will be able to define your recording file format. So you can record it into DAT, you can record it into Pico, .m, CSV, or XDF. Okay. So for now, maybe start with the DAT file. And here I can choose the root directory of my recording. So yeah, that's just put it here. Okay, users and my user folder on Windows, documents, and recordings. Okay. So you can close it. And on top of here, you can set the experiment name, the target tags, and session tags. So, and while you're changing this, the file format, so all those informations will be merged into your file name right here. So it's time, the experiment name, the participant tag, and the session tag. Okay. So experiment name. So for example, let's say PhysioLabXR example. Okay. Now, because I have a stream right here and it's actively streaming, in the recording tab, we can start the recording. So while we start recording, at the bottom of the PhysioLabXR, you can see the recording file size increasing. So this number will indicate the file size on your desk. So let's record for a while. And okay, now let's stop. Perfect. So it says my experiment has been recorded to this file directory. And I can copy this path, or I can open the directory right here. See here, we have multiple experiments recorded previously. So that's the recording in PhysioLabXR. So here we have the graphic user interface for recording, and you can specify the data format, the root directory, and the experiment name, as well as the participant tag and session tag. So the next feature I want to introduce is a replay synchronized multi-stream data. So inside PhysioLabXR, you can replay any pre-recorded data with all streams synchronized, and it's an ideal tool for testing multi-input machine learning models. So after you go to the replay tab, here is the replay interface. Inside this, you can select the recorded experiment, and it will automatically detect how many streams in your recorded experiment and how many data points in your experiment for each channel. And you can select which data type you want to simulate. So basically, the replay feature in PhysioLabXR is you record the data from an experiment, and in replay, the PhysioLabXR will simulate the same streams with the data you recorded. So in this method, you can test your pre-recorded experiment over and over again with different signal processing pipeline, machine learning model, classifiers. So you don't have to recruit participants over and over again and have someone wearing the EEG for 10 or 12 hours per day to get the real-time feedback from the participants. Yeah. So let's jump into the replay. Okay. Before we do the replay, let's stop the simulated stream from this Python script. Now the stream, you can see the label at the bottom turns red. That means the live streaming layers have for this stream has disappeared in the local network. And in the replay browse, so let's find the directory. So it's right here. And so we got this recording file. It's called PhysioLabXR example. Okay. So I select it and I open it. Perfect. So once you open the recorded file, you will get this. So in the stream names, you will see all the streams contained in this recorded file. and how many channels and time points for this stream, and the average sampling rate, as well as the data type for the stream, and how you want to simulate the stream. So you can simulate it using LSL, or you can simulate it with ZMQ. So ZMQ is a more basically ZMQ is a library that's very similar to LSL, but it's more generous. So at this point, I can start the replay. Now, once I start the replay, you can see all the streams has been started. Okay, so here, so I have to change the nominal sampling rate back to 250, and let's change the seconds of display to 10. Okay. So at this point, we just replayed the recorded streams from the simulated EEG source. Oh, so that stopped, and by clicking this button, we can start over the replay server. And in the playback window, you can see the timestamp from the local clock and the percentage of the replay and the seconds elapsed since the start of the replay. Let's go back. So at this point, we introduced how to add a stream, visualizing with different data formats, how to change the nominal sampling rates, the duration of the visualization window, how to record the data, and how to replay the data. So next, I would like to introduce the most powerful feature of PhysioLabXR, which is the scripting interface. So with this feature, you'll be able to add any customized signal processing pipeline, machine learning model into your experiments. So here's a system diagram of the script interface. So we introduced the main window, which is the main process. So it will capture the signal from different data workers, like from different lab simulator sources. Okay. And every time the main worker tick in the main window, it will forward the data into the scripting server. So inside the scripting server, the user will be able to define their customized Python script with the user-defined start, user-defined load function, and the user-defined cleanup function. And inside this user-defined loop function, the user will be able to access the most recent data being streamed from multiple data sources. And we will see an example shortly. So let's look into the code, how to define the start, loop, and cleanup function. Okay. So this is just a Python script with a class named myScript. Okay. And inside this function, you will be able to define the init function. So for example, in the init function, I can define a classifier like logistic regression. Okay? And in the loop function, I could define my signal processing pipeline and how would I like to train the classifier based on the most recent EEG signal from the device. And in the cleanup function, this function will be called at the moment we stop the script. So, for example, we can delete the predefined classifier and clean up the data buffer to prevent the memory leaking. And here the script interface, graphic user interface. So first, you can add a script. Right here, you can locate the file location of your script, or you can create a script from the scratch. Okay. Second, we can define the run frequency for the loop function and how long of the data you want to maintain in your script. Also, you can define which stream you want to capture inside the input function. And additionally, you can, so for example, you preprocess the EEG signal. In the output, you can define another stream with a different name and broadcast the stream to the live stream layer with a different name. So we will show you an example shortly. And the third feature is a runtime parameters. So you can define a customized parameters and change the parameters in real time inside the loop function. And finally, after you define everything up here, you can run your script by clicking the run button at the bottom. So let's jump into an example right here. Okay, so we have a script named example script. And what this script does is it gets the data called dummy edge channels. And for every loop function, for the most recent data, PhysioLabXR received from the dummy edge channel from the live stream layer. We want to add constant to the first channel of this signal, and we want to broadcast the signal with a different name, dummy edge channel modified. Okay. So the first thing, for this example, we don't need to define anything in the init function. And inside the loop function, we could add what input streams we want to capture. And so we can type in the input stream's name in the input box right here, and we could add. And secondly, we can define the name of your constant. So we define the parameter as a type float, and we can give it a number. And we can change this in real time during the experiment. Finally, we set the output. So after we modified the stream in real time, so in a real world scenario, this could be a filtering process. Okay? I got a like multi-channel streams input from the live stream layer. It could be EEG. So in the loop function, I want to run the real-time signal processing pipeline to remove the noise or artifacts. And we want to broadcast the signal again to the local network for other scripts. Or it could be input from the eye tracker. So the output could be the fixations or users arousal. So here we have the examples. Let's run the example. in real time. Let's have a live demonstration. So if I go to the script, I run the simulated EEG signal again with the same name before the dummy 8 channel. And inside the PhysioLabXR, we can restart the streams. So to better visualize this, let's add constant to all the signal groups. So we can visualize all the channels separately. Perfect. And in the script interface, what we can do is we could locate a script we built before, or we can create a new one. So in this case, we could create a new script, okay? Just in the recording folder. It could be anywhere. So let's call it example script. And click save. So after defining the script, we will automatically return you basically a Python script. And inside this Python script, there will be a class which is has the same name as your file, the example script. Okay. And if you don't like the text editor, so you can go to your Python editor right here. And sorry. So, or I could, alternatively, I can create a script right here. So called example script. Perfect. And I can copy the script right here. Oops. Sorry, it's time to copy the script. Format is a little bit awkward. Seconds. Or actually, I can write the script right here. Define init function and define loop function right here. And based on the example, we showed you guys. So we first we set up parameter and equal self.input and get data. So this is a function that's been defined in PhysioLabXR. So this is a helper function. And with this function, we could get the most recent data from the string with a string name. So we call it, so the name of the string is a channel. From a channel. And here we could further define the timestamps. Copilot is very smart. So timestamp input, and we get the timestamp of the dummy 8 channels. The reason we want to get the timestamp is we want the processed data has the same timestamp as the input data, the dummy 8 channel. So in that way, we could specify the output signal timestamps as we broadcast the processed signal. So in the data, 0. So what we are doing is we're adding a random number to the first channel, which is the first row of the input signal. Ramps. Okay. So we call it like your constant. Okay. So here we are at a constant to the first channel of the data. And finally, we want to send the data to the LSL outlet with the name dummy 8 channel modified. Okay. So here we could say set output, and we specify the string name, and we specify the data, and we also specify the timestamp. So if you hold your control on your keyboard on Mac, it will be out. Okay. And you can jump into the source code and look into the detail about how it creates the streams and how it broadcasts the signal into the LSL local network. Okay. So, and after every loop function, we want to clear the buffer data. So we don't resend the same samples again after we process them. Okay. So the input dot clear buffer data. Format the file a little bit. Yeah. Get some data, add a few more comments. Okay. The timestamp from the input stream on the channel. Perfect. And at the end, we could define a cleanup function. So we just simply say script start and cleanup. Okay, perfect. So at this point, we created a Rena script in PhysioLabXR. So And let's go back to the application. So in the main window, we still have the streams running. And inside the script, okay, we can, so let's find the, sorry, let's find the path of this script. So let's open it, export. So I can find the root path of this. Okay. So here's the script we just created. And it will get the data, the most recent data batch from the lab stream layer with the stream named dummy HN. Okay. And it will add a random number to the first channel with the parameter you define in the params. Okay. And it will broadcast a new stream with the name dummy H channel modified. And here we can specify the run frequency. So sometimes we may want a lower latency. So let's just say we want to run it maybe 20 times per second. Okay. And inside the input, the data we want to capture is the data stream dummy A channel. So I add it. So we can say it's dummy A channels, and it has 8 channels. And the data buffer will save the data up to 10 seconds because the sampling rate of this stream is 250. So that's why we have the 2.5K data buffer right here. And the name of the parameter is your constant. Add it. And here I could select the data type. So let's say I have a 10 right here. And the name of the output will be dummy if channel modified. And right here, we must specify the output format of the output stream. Because we are processing LSL stream without changing the channel number of the stream. So we still got 8 channels as the output. Okay. So one thing should keep in mind is we must keep the definition of those parameters and name of the output same as the name you define in your script. So for example, in the script interface, I'm adding the first row with the parameter named your constant. Okay. So in that case, in my params definition right here, I must have a parameter with name your constant. Okay. And it should be a float or integer. Okay. Otherwise, if you click the run button and in the console log, there will be exceptions that remind you, okay, you defined the parameter with runtime. You cannot add a Boolean with a float, or you cannot add a string with a float. Okay. So at this point, we click the run button right here. Perfect. Now we entered the loop function. And sorry, seems here is a small key error. And there might be some syntax error. Stop. Let me quickly debug this. Channel. Oh, so I can find the reason. Side PhysioLabXR. Let's go back to the channel. Okay. Let me quickly test. There could be some problem with the simulator. So, modify my script. Okay, that works. So my computer is a little bit um, old. Sorry for, yeah. Now we add a string back. Okay. So inside the so inside the script interface, you can change the number you add to the string. So let's pop up the script interface. And as we discussed before, basically we are modifying the first channel number right here. Okay. And you can see as we changing the parameter right here, the value of the first channel has been modified in real time. And this feature is extremely useful during the closed loop brain-computer interface development process. So you do the real-time data acquisition, and inside the script interface, you can process your data in real time and feed your processed data into like a machine learning model and outputs the feedback to the user. So here's a basic functionality for about the script interface, and for more detail, you can refer to our website. So let's go back to the presentation. Okay. Now let's look at the system diagram of PhysioLabXR. So after the user clicks the add button in window, the system will create a worker thread dedicated to receive the data within the corresponding string widget. And the worker thread then pushes the data to the visualization thread, scripting thread, and recording module. So here is an example of the experiments from our lab. So we have the eye tracking data right here, and you can change the name of the group, name of the channel, and we are adding the audio input from the participants. And we have the real-time fMRI data from the scanner. So that's experiment we did in the Zuckerman Institute in Columbia University. And we're adding several customized scripts to output the participant arousal and attention. So basically, it's doing a real-time inference for target and distractor. So when the user look at the target, the probability for the target will increase. So basically, that we're doing a binary classification on this based on the EEG data, pupil, and fMRI scan. So I know we are a little bit out of time, but to download the PhysioLabXR and run it on your personal PC, you can apply the JetBrains free educational license. So that's a very fancy Python interface, Python editor. Okay? And here's a link to download the PyCharm Professional. You can find those links in our Hackathon website. So if you go to the Hackathon website, you go to the Neureality Hackathon website, and inside the workshop section, if you click in right here, the preparation requirements, you can see how to apply the PyCharm Professional license, okay, and how to download it, and how to install Python, as well as how to clone the repository to your personal PC. And if you run it from the source, you clone the project, and then you can do the pip install with pip install -r requirements.txt or pip install -r requirements.dev.txt. So those are two requirements. So those are two requirements folder in the PhysioLabXR repository. Okay. So inside the requirements.deb.txt, it includes more packages like Matplotlib or TensorFlow or PyTorch, stuff like that, and Seaborn. So those are very big machine learning library or graphic library. Okay. So at the end, I would like to express my appreciation for everyone's participation and engagement. If you have any further questions, please join our PhysioLab Slack channel. So if you go to our official website, PhysioLabXR.org, you will be able to see a button and a link to our Slack channel. Okay. Our community is filled with future innovators eager to discuss the next generation of brain-computer interfaces. And here you can chat and share your ideas with one another. Okay. And here are QR codes you can scan as well. Hey, so I think that's everything about the session. Thank you again for everyone's participation and engagement. And see you in the hackathon next week, next Friday.